name is Johan, and this is the tutorial on removing unwanted variation from high-dimensional data. I'm being helped out today. Ramyar is here, and Sepide, and Anna, and Marie. So all of us are running the tutorial today. The general format is going to be, for the first session, the first hour and a half, I'm going to go through some slides and do some demonstrations of the RUV package. And then during the second half of the session, that's going to be a lot more hands-on, and that will mostly be run by Ramyar, Marie, Sepide, and Anna. And in those, we'll work through, again, or you'll work through a bunch of example data sets. Okay. So for this first bit, for the first session, I'm gonna go through some slides and also introduce a Shiny app. And the basic structure is going to be for roughly the first half hour or so. I'm going to give just some, I won't go so far as to say theoretical, but some general conceptual view of removing unwanted variation and the framework for RUV. Then the second 30 minutes, I'm going to talk a little bit more concretely about the RUV package that's on CRAN. And then during the final half hour, I'll demonstrate a Shiny app. So that's roughly how it's going to go. But there's a whole lot of material, and I want to adjust the pace depending on what all of you are comfortable with. So please ask as many questions come to mind. Questions are going to be very, very helpful. Okay, so I'll go ahead and get started. A couple other notes just before I really get into the main material. I wanted to point out, and I sent out an email about this, but I'm not sure if all of you got it. I sent an email to 30 people, but there's more than 30 people in the room, so I know at least some of you did not get it. Everything that I'm going to be saying today is in a Docker app. So here's where that's available. If you have Docker installed, you can run it using this command, and in particular use this tag. And if you do that, what will pop up is basically exactly what I've got running here. So I'm actually running a Jupyter notebook inside this Docker app. Okay, so everything that I do can be replicated. Similarly, there's a bunch of material on GitHub. If you didn't get the email, you will at some point want to look at this material on GitHub. I'll remind you of this before the break, but during the next session, there will be workbooks and stuff here that you'll be working with. Okay, so the way this talk is going to go for the first session, I'm going to introduce a data set. It's going to be a gene expression data set. And I should say right now, pretty much everything that we talk about today, the examples are going to be with gene expression data. But the methods should apply and do apply to several other types of data, but to keep things concrete, we're going to focus just on gene expression data. So I'm going to first go through an example data set that highlights some of the issues that we're going to be trying to solve. I'll then talk somewhat conceptually about the RUV framework for dealing with the problems of unwanted variation. Like I said, then I'll talk a little bit more concretely about the RUV package, and then I'll move on to some examples with Shiny. Okay. So here's the data set that I want to introduce. It's a gene expression data set so the original purpose was it was part of a study in which the researchers wanted to discover genes that were differentially expressed in the brains of men and women. So the study design was they had 5 men, 5 women post-mortem, and for each of these 10 people, they took 3 tissue samples. one from the anterior cingulate cortex, one from the dorsolateral prefrontal cortex, and one from the cerebellum. And then they sent aliquots, so this is a total of 30 tissue samples, 3 samples from each of 10 people. And they then sent aliquots of each of these 30 tissue samples to 3 different labs, Davis, Irvine, and Michigan. And at each of these labs, they assayed the samples with a microarray and got gene expression data on 12,600 genes. Okay. I'm going to show you what the data looks like. Here I'm going to load in a file, gender.rda. That's inside the Docker package. It's also on the GitHub. And then I'm going to look what's inside there. There's 2 versions of the gene expression data. There's a normalized version and a raw version. The normalized version has been quantum normalized using the RMA package. There's also a data frame of information on the genes and a separate data frame of information on the samples. Okay, so I'm going to look here at the version of the gene expression data that has been normalized by RMA. I'm going to call that Y. And here is just a subset of it, what it looks like. So I've got the columns are genes. And again, this is just a subset, but there's 12,600 columns for the 12,600 genes. And then there are 90 rows for the 90 microarrays. Again, 30 tissue samples, each done at 3 different labs. Okay, here's what's inside the data frame on the samples. So we've got the different cell files here, and then which patient it comes from, whether that person was male or female, the brain region, which lab it was done at, and then also, we'll ignore this, but the version of the microarray. Similarly, we've got information on the genes, and the information that we have on the genes is, gene type. Here it just shows a bunch of others, but there's also in there whether it comes from the X or Y chromosome, whether it's a housekeeping gene, whether it's a spike in control, and I'll explain more about what all of those different things are later. There's the gene symbol, there's the chromosome that it's on, and then there's also dummy variables for whether it's a housekeeping control, a spike in control, or a positive control. Again, I'll explain what all these controls are later, but this is just so you can see what's in the data. Okay, so here I'm going to load the RUV library, load ggplot, define a couple other things mostly related to ggplot, and once I do that, I am able to make this plot. Okay, this plot is going to tell us a lot about basically the problem that we're trying to solve. So I have here, so this is a principal component plot of the data matrix that I showed you, the Y, the gene expression data. And by color here is the brain region, and then the plot symbol is the lab that it was done at. And remember, we took the same 30 tissue samples and sent them to 3 different labs. So ideally, we should have basically gotten back 3 sets of the same data. But what we actually see in the data is that the biggest source of variation is by lab. So we have all of the Davis samples are over here, all of the Michigan samples are over here, and all of the Irvine samples are down here. This is an example of what we call unwanted variation. You'll also hear this referred to as batch effects. Okay, so different labs, different batches. Again, this data has already been normalized by RMA, and yet the biggest signal in the data by far is still the brain, or the lab, excuse me. Did I see a question? No? Okay. So this is the problem that we're trying to solve. One thing that you might conceivably do, and people sometimes do, is say, okay, well, the first couple of principal components are just batch effects from the lab. So how about I just regress out a couple of the first principal components and then take whatever is left. So residualize to the first few principal components. Here I've done that, and specifically I've removed the first 5 principal components. The good news is that the batch effects are largely gone. The bad news is there doesn't seem to be much biological variation left either. We don't see, for example, any clustering by brain region. And now just as a bit of a teaser, here is that same data set but adjusted using one of the RUV methods. And after that adjustment, you see that we have all of the cerebellum clustering here, and all of the cortex is clustering here. Okay? So again, this is just a bit of a teaser. I'll show you how we got this as we go through the slides. Any questions before I go on? No? Okay. So I'm going to talk now about the RUV framework. And so I guess I should say that there's not just a single RUV function, not just a single normalize it function. There's a bunch of different ways that we can approach a data set like this and work to remove the unwanted variation. But central to all of the methods is going to be notions of negative controls and also replicates. I'll explain what those are as we go on, but know that what is the key core idea to the RUV methods are these negative controls and replicates. Okay, so what I'm going to talk about just for the next 5 to 10 minutes, I'm going to introduce a statistical model. I'm going to explain the notion of negative controls, and then I'm going to explain replicates and how all of these are used to remove unwanted variation. So here's the model. Y again is the gene expression data, and this is an M by N matrix. M is the number of microarrays, and N is the number of genes. So it's a short, fat matrix. X is going to be the factor of interest. So in our case, X would be gender. And beta is the coefficients for the factor of interest. So we've got coefficients for each genes. And in our case, in the differential expression study, this is what we're trying to estimate. What is the effect of gender on each of the genes? W is the unwanted factors. This will correspond to things like batch, but also things that might be varying within batch, such as changes in temperature, changes in the composition of the atmosphere, many various things that can affect the gene expression data that are not of interest. Alpha is the coefficients, and then we also have a just straight up plain pure random error term. What makes all of this complicated is that this W, the unwanted factors, things like changes in temperature, changes in composition of the atmosphere, these are very often either just totally unknown or we don't have any measurements on them. Okay? So it's basically, it looks at first basically just like a straightforward regression model, linear regression model, and it is, but with the important added complication that this W is unknown and we need to find some way to get it from the data. Okay, and I should point out that this model, at least in the context of genomics, first came from Leek and Story and Stagel in 2007 and in 2008. Okay. So now I'm going to talk about what are negative controls. Negative controls are going to be features, or in our case, genes of the data, that are unaffected by the factor of interest and that are affected by the unwanted factors. Okay? And so one example of these would be spike-in controls. So on a microarray, you have spots for, say, about 10,000, 12,000 genes. There's also some spots that don't measure RNA from human genes. They measure RNA from bacterial genes, and this RNA is spiked in during the course of the experiment. Okay. In the microarrays that we're dealing with, there are 33 probes that measure these spiked in bacterial RNAs. Now, the bacterial RNA that's spiked in at a fixed known quantity, and typically what's this used as is a quality check. So we know how much of the bacterial RNA got spiked in. We look at the probes associated with those spikes, and we check to see that it's basically what it should be. And if it's not, we throw out that microarray. So that's what the spike-in controls are on a microarray. We're not going to use them just to identify bad arrays, though. We're going to use them to actually clean up the data. So here's the idea. Suppose that we have a number n sub c of negative controls. In this case, we can think of spike-in controls. So 33 in our case. And we're going to let y sub c be the m by n sub c submatrix of y containing only the columns of the negative controls. Okay. And we're going to define beta sub c, alpha sub c, and epsilon sub c similarly. So this is going to be the coefficients for gender just on the spike-in controls, the coefficients for the unwanted factors just on the spike-in controls, and so on. Okay, so since y sub c is a submatrix of y, the same model applies, but again, just subsetted to the negative controls. And then what happens is this beta sub c is equal to 0 by assumption. These are, so we assume straight up that the spike-in controls are not affected by gender. That makes sense. They're spiked in externally. It shouldn't matter. The gender has no impact on these spots on the microarray. Okay, so this is equal to 0 by assumption, which means this term disappears, which means that we now have this, which looks just like a factor analysis model. And so we can perform a factor analysis on the y sub c, and that will give us an estimate of w. Okay, so big picture here, by assuming that we have some negative controls, by assuming that some of our genes, or in this case spike-ins, are unaffected by the factor of interest, are unaffected by gender, what we're able to do is we're able to run a factor analysis on just those genes, or just those spike-ins, and we are then able to estimate the unwanted factors from the data, from the negative controls. So the key assumption that goes into all of this is that the regression coefficients, the beta sub c's, for the unwanted factors, or excuse me, for the negative controls, are equal to 0. Okay. This gets a little bit subtle, so I want to stop and highlight it very clearly. What we're assuming about the negative controls is effectively that they are not affected by the factor of interest, which is different than saying that they're uncorrelated with the factor of interest. Okay? And the reason that so you can imagine a situation where the unwanted variation is itself correlated with the factor of interest. Perhaps the study was designed somewhat poorly, and as a result, some of the unwanted factors or the batch effects end up being correlated with the biological factor of interest. In that case, the negative controls will also be correlated with the factor of interest. Okay? But it's not because the factor of interest has an effect on them, it's just that they're correlated. Okay? The flip side of this is that we can't discover negative controls de novo from the data. We can't just go through the data and say which genes are uncorrelated with the factor of interest. Okay, I'm going to call those the negative controls. You can't do that because, again, the negative controls might very well be correlated with the factor of interest. what defines the negative controls and how we have to choose the negative controls is that they are things that we know a priori are unaffected by the factor of interest. Okay? So in the case of the spike-in controls, that works because of the fact that we know that these are spiked in exogenously. They're not affected by gender. All right, I'll pause in case there's any questions up to this point. Okay. One other quick note before I move on. Negative controls don't need to be perfect. I'll discuss this a little bit more as we go on, but often sparse, meaning that the vast majority of the genes that we call negative controls are unaffected by the factor of interest, or even in some cases just that the effect is 0 on average will do. Whether or not this is the case will depend a little bit on the exact RUV method that you're using, and I'll highlight that as we go on. But the reason I want to point it out now is it feels like this negative control idea is very limiting. You need to know which genes are unaffected by the biology that you're interested in. It doesn't need to be perfect. Okay. So I've shown that the negative controls can be used to identify W, the unwanted factors. We still need to, however, estimate the alpha so that we can remove all of the unwanted variation. There are several ways, once you have the W, that you can go on to also estimate the alpha. One way is to have a known factor of interest. So in our example, we actually know what the factor of interest is. It's gender, and we're trying to estimate the effect of gender. Okay. In other examples, what we're calling the factor of interest might not actually be known. An example would be if you're doing a clustering analysis, you have a bunch of tumor samples and you want to discover tumor subtypes. In that case, the factor of interest would be the tumor subtypes. You don't actually know what those are. Those are to be discovered. Okay. But at least in the case where you do have a known factor of interest and you're doing a differential expression analysis, then you have the X, you have the W, or you've estimated the W, and you can just do linear regression and you're good to go. In cases where you don't have a known factor of interest, other things that you can do is, in some situations it makes sense to assume that your unwanted factors and your factor of interest are independent. That is justifiable when the study design is particularly good. Another thing that you can use is if you have gene-wise covariates. So you have information on the genes such as their GC content, their length, various things that might be related to how they're influenced by the unwanted factors. I'm not going to talk very much about these, but the last thing that you can do is if you have replicates, meaning in the data set that I showed, each of the 30 samples were sent off to 3 different labs. So each of the tissue samples, there's 3 replicates of it. And we use these replicates to help identify the alpha. And I'll show how in just a second. Okay, so suppose Y1 and Y2 are 2 rows of our gene expression matrix that are replicates. So say Y1 was done at Davis and Y2 was done at Michigan, for example. Okay, then just subsetting to that first row of the gene expression matrix, the model is still that the first row's gene expression is equal to the factor of interest for that row, for that sample, times beta, plus the unwanted variation, and so on. And then same for the second. But then critically, and this is the key part, is that since these are replicates, the biology in both of the two is the same. So we know that the biology in the first and the biology in the second is the same. X1 is equal to X2. So what happens is if we subtract these 2 replicates from each other, just algebraically, this is what we get. And importantly, we have this X2 minus X1 here, which we know is going to be equal to 0. because we know that in the two replicates, the biology is the same. So a similar thing happens as with the negative controls. That term disappears, and we're left with that the difference between the two replicate arrays is equal to a difference in the unwanted factors times the alpha plus this difference in the pure errors. Now, if we've got a whole bunch of replicate differences, and we do in this data set, we can take a whole bunch of these replicate differences, run factor analysis on them, and from there we can get the alpha. Okay, so let me just back up a little bit and look at this big picture. So I've talked about negative controls and replicates and how both of these can be used to identify and estimate aspects of the unwanted variation so that it can then be removed. Specifically, the negative controls, these are genes where we know that the beta is equal to 0, and these allow us to identify W, the unwanted factors. Replicates, these are array differences where we know that the biology cancels out, and those allow us to identify the coefficients of the unwanted factors. Okay. And here, this is the teaser plot that I showed you a little bit ago. This is doing exactly what I just described. So here I am, here I'm running the RUV3 function, which does pretty much what I just described. It takes as input the gene expression data, the Y. It takes a replicate matrix that I'll describe in more detail in a minute that describes the replicate structure in the data. What is a replicate of what? In this case, basically anything that was the same patient and the same brain region, that gets treated as a replicate. And then also you specify what the negative controls are. In this case, it was the spike in controls. And then there's also this K term here that tells you how many principal components to use. Okay. Or in other words, how many factors, unwanted factors to remove. Okay, so that is what this function does. Okay, I'm going to, for the next half hour or so, I'm going to be talking a little bit about the RUV package a little bit more concretely, what functions there are, how we can use them to adjust our data. Before I do that, I'll pause for just a second if there's any questions on the basic ideas of negative controls and replicates as ways to adjust your data. Excuse me?
I mean, do you need both the negative control and replicate information?
Yeah, so do we need both negative controls and replicates? Very good. So yes and no. You will always need some notion of negative controls, no matter what. If you're doing something like a differential expression analysis, you can get away with using just negative controls. You don't need replicates. Because again, I mentioned that the negative controls, that can get you your estimate of W. And if you're doing a differential expression analysis and you already have a known X, from there you can just do a linear regression, regress Y onto X and W. So for differential expression analysis with a known factor of interest, all you need is negative controls. And that said, I also made the comment that the negative controls need not be perfect. So some of the methods that I'm going to show here in a minute are fairly robust to situations where, say you've got 1,000 genes that you call negative controls, and say maybe 5 to 10% of them actually are affected by the factor of interest. Some of the methods that I show are pretty robust to that. And so to take the extreme case, if you're doing a differential expression analysis in a case where you think that only a handful of genes in the entire genome are affected, you can even go ahead and use everything as a negative control, and it will end up working out. you have a question? No? Okay. Yeah, I thought I saw a hand behind you, sorry. Any others? Yeah.
Is this method basically a kind of factor analysis, is that right?
There are factor analyses that are part of it, yeah.
And how do you select the factors?
Say,
How do you select those factors, to put in the model? For example, we have 4 or 5 factors,
mm-hmm,
right? So we have the script plots,
mm-hmm.
for example, to how many factors we can choose and put in the model.
Ah, okay.
This method, how do you select the factor? Just put it in manually, just choosing by yourself?
Ah, okay, so very good. So, yeah, here, for example, in the RUV3 function that does this analysis, there's this variable k here, goes off the screen, but it's actually equal to 10. So what actually happens in here is the factor analysis is done just by a singular value decomposition, and that k equals 10 says take the first 10 factors.
And you select the two most important ones in this.
The 10 most, yeah. And I'll mention this a little bit more as we go on also, but some of the methods are quite robust to the number of factors that you include. And in fact, you can actually essentially take all of the factors and it will work out all right. But yeah, so in general, all of the factor analyses that these methods do are just a singular value decomposition. And in some cases, there's a tuning parameter, which is the number of factors to keep. In other cases, it actually selects all of them. And, well, as you'll see, works out pretty well in that case too. Very good. Yeah? Ah, okay. So that's a really good question. So the question, just to say it back, is tell me if I'm saying it right. We're basically learning the nature of the unwanted variation via the replicates and via the negative controls. And in particular, the replicates are the replicates across these 3 labs. And so the question is, if we have more data coming in the future that comes from these 3 labs, can we apply the same normalization to this data that's coming in the future? The answer is a qualified yes, as long as these labs keep doing more or less what they've been doing in the past. You'll be able to adjust for the unwanted variation coming out of these labs. So in principle, so for example, say that an 11th person entered into this study, but this 11th person was only assayed at Davis, not Michigan, not Irvine. You would be able to adjust that person using all of the other data from the replicates that you have. And in fact, I think Ramyar will talk about this when he goes through his example in the second session. You can actually get by with a fairly, actually I would say surprisingly small number of replicates. So even if you have maybe just 10% of your data as replicates, you can end up getting a very good adjustment. Very good question. Any others? Okay, I'll go ahead and move on then. Okay. So I should say that there are a couple RUV packages. The one that I'm going to be focusing on today is the one that's on CRAN and is just called RUV. There are 2 others that are on Bioconductor. One is called RUV Normalize. This has some earlier versions of the methods that are based on replicates and also some methods that are applicable when you're willing to assume that the unwanted variation and the variation of interest are essentially independent. And then there's another package on Bioconductor called RUV-Seq, and that is specifically tailored to RNA sequencing data. So there are multiple packages. Again, the one that I'm going to be focusing on today is the one on CRAN, just called RUV. Okay, so what I'm going to do in this next section of the talk is I'm going to give an overview of some of the main functions that are in the RUV package. And then specifically, these are broken down very broadly into what I'm going to call regression methods and global adjustments. Let me pause just for a second to say what these are. Regression methods, that's going to be methods that do essentially a differential expression. You have a known factor of interest, and you want to figure out what the effect is of that factor of interest on a whole bunch of different genes. And so the goal of the analysis is to get these effect estimates for each gene. Okay? So all of these regression methods, they output effect estimates. There's global adjustments. These are harder to do, but also in some sense more useful. A global adjustment is you take your gene expression data, you run it through a global adjustment method, and you end up with a new gene expression matrix that you can do whatever you want to. Okay? So again, global adjustment is in some sense more useful because you can use it for a whole bunch of different purposes, not just doing a differential expression analysis, but statistically and conceptually it's a little bit harder to do, and in particular the global adjustments are what typically require the replicates, whereas the regression methods require just the negative controls. Okay, so again, I'm going to give an overview of the functions, I'm going to discuss the regression methods, and I'm going to discuss the global adjustments. And then after that, I'll move on to the Shiny app. Okay, so overview of the functions. Again, there are global adjustments and there are the regression methods. The global adjustments, there's basically two methods, RUV1 and RUV3. RUV3 is the one that I showed you that takes as inputs negative controls and replicates. RUV1 I'm not going to really talk about today, but this makes use of external information that you might have about all of the different genes. So things like their GC contents, their length, and anything else that might affect how unwanted variation affects them. Okay, so those are the global adjustment methods, and again, today I'm just going to focus on RUV3. And then there's the regression methods, and all of these do essentially the same thing, but in slightly different ways that give them different strengths and weaknesses. So all of these use the negative controls to come up with an estimate of the unwanted factors. So just like I showed you earlier in the slides, I took the negative controls, I did a factor analysis on them, and then I have my estimate of W. What I described, just as I described it, is RUV2. Okay? Take the negative controls, do factor analysis on them, call that your W, and then do a regression. That's RUV2. RUV4 does a very, conceptually, high level, does a very similar thing, except that it is far more robust than RUV2 to misspecification of the negative controls. So if you do RUV2, and in fact, say 10%, 20% of your genes actually are affected by the factor of interest, RUV2 will break down. RUV4 very likely will not. Okay? Both with RUV2 and RUV4, you need to pick a number of factors to include in the model. And that's the K that I was mentioning just a few minutes ago. With RUV, this is RUV inverse and RUV R inverse. With both of these, you do not need to select a number of factors to include in the model. It actually includes all of them. And I won't go into the math of it, but it actually starts to become something more like a mixed effects model. Okay, these two, like RUV4, are much more robust to misspecification of the negative controls. Okay, so those, what I just showed you, those are the core adjustment routines. Then there's also some helper functions. Specifically for use with RUV3, there's a matrix that produces the replicate matrix. The replicate matrix describes the replicate structure, and I'll show in a minute how to use that. There's also a function called collapse replicates. I may or may not go into this, but basically it's for eventually after you've used your replicates to estimate the unwanted variation, you might not really have any use for them as replicates anymore, and you might just want to average over all of the replicates. So, if you have one from Davis, one from Michigan, one from Irvine, there might no longer be a point in keeping all of them as different arrays. You just want to average over all of them. If you do that, then you also want to, in some sense, average the metadata. This takes care of that. For the regression methods, there's a function that summarizes the outputs of the RUV2, RUV4, RUV inverse, and RUV R inverse methods. So this is a very common and useful function. And then there's these 4 other functions which you probably won't use too often but might use occasionally, so I have them listed there. I won't talk about them today. There are a bunch of plot functions. Some are somewhat general and are useful for mainly for diagnostics of how much unwanted variation there is in a data set. I'll go through some of these in a minute. They're also integrated into the Shiny app. And then there's also some plot functions that are specifically for the regression methods. Functions to make histograms, empirical CDFs of p-values, various things that I'll come back to. Okay, and then there's a bunch of functions that are defined in the package that typically you don't use. So I'm just going to list them here so you know that these are things that you typically won't use. And I especially want to highlight this function getK. GetK is a function that might be useful for RUV4 in telling you how many factors to include in the model, how many of the factors from the factor analysis to include. But typically, when you're doing RUV4 or RUV2, you'll want to choose that K by hand. In fact, one of the main strengths of RUV4 and RUV2 is allowing you to choose that K by hand. The only reason that this is included in the package is for situations where you do need to choose a K, but it just is not feasible to do it by hand. For example, if you're doing some simulation studies. But in general, I want to point out that this really is not something that you want to use. Okay. And then there's also the Shiny app, which is built into the package, and that's RUV Shiny. And I'll come back around to that. Okay, so I'm going to start with the regression methods, and I'm basically going to just work through an example analysis after I show you here some of the basic syntax. Okay, so again, there's RUV2, RUV4, RUV inverse, and RUV R inverse, and they all do basically the same thing. They all estimate a W using the negative controls, and then regress Y onto X and W. So all of them, you need to give them the gene expression data, the Y, the factor of interest X, an index variable for which of the genes are the negative controls. For RUV2 and 4, you need to specify how many factors you want out of the factor analysis. And then there's some extra optional variables too that I will mention here in a second. Here is a summary of the different arguments. I'm going to walk through them one by one, so I'll just skip that table. Y again, that's the expression data. It's an M by N matrix where M is the number of rays, N is the number of genes. Typically, at least with gene expression data, you'll want to log transform the data. And now this might be a little bit counterintuitive, so I'll pause on it for a second. So typically with gene expression data, you will want to log transform it. But beyond that, it's best not to do any further preprocessing. In particular, it's best not to do any quantile normalization. The reason for that is, so you're gonna designate some genes as negative controls, and the assumption on these negative controls is that they're unaffected by the factor of interest. If you do a quantile normalization, and a very large number of the genes are affected by the factor of interest, and the quantile normalization is keeping the overall distribution fixed from sample to sample, then when some of the genes that are affected by the factor of interest all go up, then the quantile normalization will essentially force even the negative controls to go down. Okay? So in other words, quantile normalization can actually induce a signal of interest into your negative controls. So that's one thing to be aware of. So in general, again, it's best not to pre-process the data in any way other than a basic log transform for gene expression data. X is your factor of interest. That might be something like gender or brain region. You don't want to include the intercept in X. That's automatically included elsewhere. And just as a rule of thumb, if you've got several factors of interest, say you're interested both in gender and brain region, a rule of thumb would be the fewer factors that you include in the RUV function call, the better. The basic reason for this is that the more factors that you include in your X matrix, that's the fewer degrees of freedom that you leave left for estimating W. And so what typically tends to be better is just to repeat the analysis a couple times, once for each factor of interest separately. Z is any other covariates that you want to include in your model. So these things might be known batches, known labs, things like that. The intercept is included automatically in Z, although you can remove it if you want. And then there's a similar rule of thumb for what to include in Z. And much like X, the rule of thumb is the less you put in it, the better, actually. Again, the reason is the more you put in the Z, the fewer of degrees of freedom that are left available to estimate your W. And in practice, it's actually often the case that your estimated W from the factor analysis actually does a better job at capturing the unwanted variation than including a dummy variable for known batch. And that seems kind of surprising at first, but when you think about it, it's maybe not so surprising. So if, for example, the reason that you have unwanted variation in your data that varies from batch to batch is because of, just hypothetically, as an example, temperature variations between the different labs, well, there can also be variations within the lab in temperature, and that won't necessarily be captured perfectly just by including a dummy variable for batch, but the factor analysis will pick it up pretty well. Okay? So in practice, it often turns out that it is better, even if you know batches, it's better to leave them out and let them get pulled into your estimate for W. There is one important exception, and that is when you know of some factor that likely affects only a very small number of genes, and importantly, also these genes it affects are also very likely the genes that may be affected by your factor of interest. So an example would be, say you want to do a differential expression with regard to some disease. So X is the disease state, and suppose that the disease affects only a small number of genes, and then say also that you have some information on who is taking a drug that also affects this small subset of genes. Okay? In that case, it probably would be good to include the information that you have on who is taking the drug. The reason being that since that factor affects only a small number of genes, it very likely won't get picked up in the factor analysis, but it is something that you very much want to adjust for. Okay, let's see. Eta, that is gene-wise covariates that are likely associated with the unwanted factors, things like GC content, gene length, etc. This is just included for convenience in the differential expression functions. It's entirely equivalent to pre-processing the data with RUV1, which is something that I said I was going to mostly skip over for this talk. Yeah, so I'm going to skip over that. And then there's the control variable, and this is one of the most important things to think about when you run an REV method. It's crucial to the success of the method. And so ideally what you want is genes that are unaffected by the factor of interest, that are affected by the unwanted factors, and ideally also they'll be somewhat representative of all of the other genes, meaning that, for example, they have a similar range of expressions, and also ideally that they're not affected by their own unwanted factors. So for example, in the case of the spike-in controls, the spike-ins get spiked in separately than everything else. Sometimes someone might spike in a little bit too much or a little bit too little, and so the spike-in controls will themselves have sources of unwanted variation that are not representative of everything else in the microarray. Okay? So that makes them somewhat unideal. But the three things that you really want out of your negative controls, they're unaffected by the factor of interest, they are affected by the unwanted factors, and that they're representative of the other genes. Okay? And I said this earlier, but I want to say it again. Negative controls, they can't be automatically discovered from the data, at least not in any sort of naive way. You can't just go through all your genes and look to see which of them are uncorrelated with the factor of interest and call those negative controls. Again, the reason is it's entirely possible that negative controls are and should be correlated with your factor of interest. That will happen whenever the unwanted factors themselves are correlated with the factor of interest. Okay, and then something else I've already said before, the negative controls don't need to necessarily be perfect, and some of the REV methods are quite robust to varying degrees. So if, for example, 10% of your genes actually are somewhat affected by the factor of interest, for at least some of the methods, that's going to be fine. Okay. There's the parameter k, that's for RUV2 and RUV4. That's the number of unwanted factors to include in the model. And when you want to do this, so there's a special situation when you want to make use of this k. And that is going to be when the negative controls, when you suspect that they do contain at least some biology. Okay? In that case, keeping k small will reduce the risk of over-adjusting. So if you think that your negative controls maybe have a little bit of biology in them, and you're concerned that you might remove that biology along with everything else, one thing that you can do is do RUV2 or RUV4 instead of RUV inverse or RUVR inverse, and keep k small. And then that way you'll only remove one or two factors, and they will ideally not be the ones that have any biology in them. Okay? And I've said this before also. Basically, if you are doing RUV2 or RUV4, it's most likely because you have this concern. And if you have that concern, it's best to choose k by hand using various diagnostic plots, which I'll show in a minute. And you don't want to use the get k function that's included in the package. Okay, so here's a big table that compares the 4 different regression methods and their strengths and weaknesses. I won't go through all of it. It's more of a reference slide that you can go back to. But just to highlight again, RUV2 and RUV4, those both allow you to set k, the number of factors to adjust for. And typically, you'll most want to do that when you are concerned that there's some biology in your negative controls. RUV inverse and RUVR inverse are methods that are robust to the choice of the number of factors, and so they just by default use a very large number of factors, so you don't need to pick a k. Both RUVR inverse and RUV inverse are also fairly robust to misspecification of the negative controls. So if there is some biology, as long as there's not too much, both of those will probably be okay. And as a first option, very often the thing that you'll want to use is the RUV-R inverse. That one will work in most situations. So as I have it here, it has a good compromise of features. Yeah? Oh, good. Let's, where is that? This one is right here, yeah? Okay, so RUV4, much more than RUV2, RUV inverse, or RUV-R inverse, is sensitive to negative controls that are not representative of the rest of the genes. An example of that is the spike-in controls. As I mentioned, like sometimes the spike-in controls will have their own unwanted variation in them because you spike in a little bit too much, you spike in a little bit too little. RUV4 is quite sensitive to that. So as a general rule of thumb, I would not use RUV4 with spike-in controls.
All the other RUV inverse, RUV-R inverse, you also said it's some more sensitive to non-sensitive.
Yeah. So RUV inverse, RUV-R inverse are somewhat sensitive. RUV4 is much more sensitive. RUV2 is nearly insensitive to this.
If we're using spiking controls, we should use RUV2.
Generally, that's what I do. Yeah.
This spiking control is only for the microarray spiking because they now have kind of RN6 spiking, similar way.
What's that?
RN6 spiking.
I didn't quite catch it.
Spiking for RN6.
Oh, yes, yes, like the ERCCs.
Yeah, and all the four the same way.
Yeah. Mm-hmm. Yes. Uh-huh. Okay. And I've mentioned a couple times that RUV4 inverse and R inverse are more robust than RUV2 to violations of the negative controls. So here, it's a little bit cryptic, but I'm still going to put it up there, what exactly that robustness is. For RUV2 to work right, you really need that the negative controls are all unaffected by the factor of interest. You actually need these beta C's to be 0. For the other methods, you need something that's weaker. It's kind of weird. It's something that is clearly a weaker constraint, but it's also somewhat hard to interpret. And what you need is for the effects within the negative controls to be more or less orthogonal to the effects from the unwanted variation. Again, it's something that's pretty hard to interpret, but it's also something that is very clearly weaker than assuming that the beta C is 0. Okay. Okay, so here I'm now going to work through an example analysis. I'm going to, and here's how it goes. I'm going to use RUV-R inverse to do a differential expression fit. I'm going to pass in Y, that's the gene expression data. I'm going to pass in the gender dummy variable. That's my factor of interest. And then I'm going to pass in the spike-in controls. Those are my negative controls that I'm going to use. That returns a fit, so I'm going to save it in this variable fit. And then I'm going to use the function RUV summary, which will take that fit and make a nice summary table out of it. So that again takes, I need to pass the expression data back, the fit variable, and also any information that I want to include in the summary about the samples, and any information that I want to include in the summary about the genes will go there. And then what comes back is a whole collection of useful things, but in particular there's one variable C, and that tells us for each of the genes, here we have genes as all the rows, we have p-values FDR adjusted p-values. Here we have the log fold changes, estimates of the variances, various other pieces of information, including this information that I passed in, such as the gene names, the gene types, etc. Okay, so that's an example analysis, just a couple lines, and you have a very nice clean summary of the analysis. You can also take that summarized fit and pass it to some of the plotting functions. So here I've passed the summarized fit to a function called RUVHist that makes a histogram. I want to stop on this for a second and also just point out the value of making these histograms. So when I do a differential expression analysis, in the absence of any unwanted variation, this is actually the kind of histogram that you'd want to see. Most of the genes are going to be null, and the distribution of the p-values for the null genes should be a uniform distribution. So when I see this here, this is more or less a uniform distribution. This is being formed by all of the genes that are not affected by gender. And then ideally, you'll also see a spike here down at 0. These are the very small p-values that come from the genes that actually are affected by gender. Okay? So ideally, this is the kind of p-value histogram that you'll see. And after the adjustment, this is what it looks like. I'll show you the histogram from an unadjusted analysis in a few slides. Okay. Another function that's included that is sometimes useful, instead of a histogram, there's an empirical CDF of the p-values. You'll notice that you can't actually see the spike at 0 in this empirical CDF, so you can also do a power transform of the axes, then you can very clearly see the spike at 0. Why might you want to do an ECDF instead of a p-value histogram if you have to go to this trouble? Is basically that it's often very helpful to look at the ECDFs broken down by the gene types. So here I have, for example, let's see, so the yellow are the spike in controls, and you can see that they have something that is somewhat uniform. They actually are a little bit right shifted. The housekeeping genes, those are nearly uniform, which is what we would want. And then these genes, these are, let's see, the purple are genes from the X and Y XY chromosome, and the blue are genes just from the Y chromosome. These are genes that you would most naturally expect to be affected by gender. And in fact, you can see that their p-values are very much left shifted. Okay, so by looking at this and knowing what the different gene types are, you're able to fairly quickly diagnose whether the analysis looks reasonable or not. Another plot that is very useful is a plot that counts the number of top-ranked positive controls. So a positive control is going to be any gene that you suspect may be affected by the unwanted factor. In our case, we could take all genes that are from the X and Y chromosome and treat these as positive controls. These are the genes that we think are probably most likely to be affected by gender. I'm going to skip to the next slide, which blows up this slide a little bit, subsets it. Okay, so what I actually have here is on the x-axis, I have the number of top-ranked genes. So I've taken all the genes and I've ranked them by p-value. And so what I'm doing here is I'm looking at, say, here I look at 10. That means that if I'm looking at the top 10 genes ranked by p-value, and then I count how many of those are positive controls, in this case, how many of them come from the X or Y chromosome, the answer is 9. So this is a pretty good sign that the adjustment is helping because in the top 10 genes of this analysis, 9 of them come from the X or Y chromosome. 9 of them are positive controls. Also included in this plot, kind of hard to see, there's this faint diagonal line here at the bottom. That is in some sense kind of a null distribution line. If you took all the genes in the analysis and just randomly shuffled them, and then asked what is the expected number of genes that would be positive controls, if, for example, you looked at the top 40, it would be here, about 3. Okay? So this is essentially the null distribution line of what this graph would look like if the adjustment was completely awful. Okay. This general type of plot is probably one of the most useful diagnostics that you can do after running a differential expression analysis. I can't emphasize enough, and will emphasize a lot after the break, that positive controls are very, very useful because whenever you do any sort of analysis on real data, there's going to be various things that you need to twiddle, work out, different negative controls that you might try, different values of k's that you might try, and you're always going to be a little bit in the dark in real life. And having some positive controls to help guide you, help let you know whether the analysis actually looks better or not, is very, very helpful. So this is probably one of the most useful plots that you can make. Okay, this is a projection plot. I won't go into too much detail on what this is, but very, very generally, the x-axis is in some sense the most important linear combination of the unwanted factors. The y-axis is the estimate of the effect, so the log-fold effect. And a little bit more specifically, the estimated log-fold effect is the vertical distance, so I should say each dot is a gene. This is one gene here, and the estimated log-fold change for this gene is the vertical distance from this dot down to this black line. RUV2, RUV4, and all the other methods, all are essentially different ways of estimating this black line. And they all do something slightly different by figuring out, so in this case, the greens are the negative controls. Essentially, there are different ways of fitting this black line to these negative controls. In any case, you can use a plot like this basically to tip you off if something looks really wrong. So for example, if this black line looked something like this, just was not fitting the green dots at all, you'd know that something was wrong. And that does occasionally happen. Here is a volcano plot. So on the x-axis is the estimated log-fold change. On the y-axis is the minus log p-value. I'll skip that one. Okay. In any analysis, you're going to want to ask yourself, did we help? And as I've mentioned, the p-value histograms, the empirical CDFs, and especially these rank plots are very helpful in letting us know if we've helped or not. Here's some code to compare p-value histograms and these rank plots before and after adjustment. On the left here is the p-value histogram of an unadjusted analysis. So this is basically what you would get in terms of p-value histogram if you ran lemma, or it will look pretty much the same also if you just ran a straight up ordinary linear regression. As you can see, the bulk of the genes, their p-values are very strongly shifted towards 1. They don't look uniform at all. and the spike is lower here than it is over here. And you can also see that in this plot of how many positive controls you get at the top ranked genes, you don't get as many positive controls in the top ranked genes as you do after adjustment. Okay, so making plots like these are very, very helpful in letting you know whether the adjustment is helping or not. Might be a good time to pause for questions again, if there are any. Yeah. Yes. The x-axis is the full change, yeah?
So since everything is below 0.5, so there's nothing different. I mean, this is it's the same or similar presentation of the volcano plot for the RNA expression analysis, right? And so the effect, I mean, the outcome of your RUV method seems to shrink everything to the middle.
What makes you say that it shrunk?
Yeah, because there's no, should be some genes that have very large difference.
Well, here's a couple.
They're only very little of that.
Well, these ones, for example, have log-fold changes of over 2.
Yeah. Yeah, but typically, if you have 12,000 microarray probes, you only get several, maybe less than 10 different genes.
I don't know. In some sense, this is a biological question, because what we're looking at is the differences in gene expression in the brains of men and women. And so I suppose the biological conclusion is that there's a couple of genes that are very different between the brains of men and women. Some of them that are a little bit different, but clearly statistically significant, and the vast majority are not different.
So you're saying that this is not because of adjustment?
No, But it's good to be skeptical. I like it.
Can I comment on that more?
Yeah.
We are going to actually show them in the next session that we do adjustment, but we see more differential expressions after adjustment. It really depends on what sorts of coverage of question you have. For example, if you have treatments after versus not treated, it will not be a huge change in genes that have differential expression. But if it is like, for example, this one, only genes related to gender will come up, and obviously, we'll show you in the class.
Another thing I can do, so I'm going to start showing the Shiny app in a second. When I show the Shiny app, I can show you a differential expression with respect to brain region instead of gender, and there you can see that there's a lot. Before I get to the Shiny though, so I did an example analysis of a differential expression. Now I'm going to do global adjustments. And again, I'm going to basically just look at RUV3. And notice that in comparison to the regression methods, where it was 4 methods that more or less do the same thing, but with different strengths and weaknesses, RUV1 and RUV3 are very different from each other. RUV1, again, is the one that uses external information on the genes, uses gene-wise covariates. I'm not going to have time to talk about that one today, but I'm going to talk about RUV3, which uses replicates. Okay, so I'm going to skip over that. So again, RUV3 requires replicates. It requires that you create what we call a replicate mapping matrix. That's this M here. That describes the replicate structure. So the arguments are Y, the gene expression data, M, the replicate mapping matrix that describes the replicate structure, the negative controls, optionally how many factors you want to adjust for, and then also optionally whether or not at the end you want to average the replicates. Okay, so this mapping matrix, let me just define it briefly. It basically maps observations, so the rows of Y, the microarrays, to replicate sets. So in the case of this data set that I've showed you, we have 30 tissue samples and we have 90 microarrays. Each tissue sample has 3 replicate arrays. And so what we're going to say is we've got 30 replicate sets. Each tissue sample will be a replicate set. And in each of these replicate sets, there will be three arrays. Okay, 3 observations. So the way this matrix is defined is the ijth row is equal to 1 if observation I is in that replicate set, or is in replicate set J, and it's equal to 0 otherwise. Okay. And as a reminder, any variation within a replicate set is assumed to be unwanted. So if we've got 3 replicates, we want them ideally to all look the same. Any variation that we see between the 3 we're going to treat it as unwanted. Okay. There's a function in the package that creates these matrices for you. It's called replicate.matrix. You pass it something that gets converted to a data frame. And then what happens is, so say you pass it a factor, and then it gets converted to a data frame that has 1 column that is that factor. Every observation with identical rows in this data frame is going to be taken to be replicates. So as another example, say that I passed in a data frame that had 2 factors, and let's say that the 2 factors are brain region and patient. Then what will happen is any observation, or any 2 observations that have that are marked the same in terms of the brain region and the patient will be treated as replicates. Okay, so that's the way that this function works. I will skip that. Okay, and so here's the first example. I'm going to use the spike-ins as the negative controls, and I'm going to use the technical replicates as the replicates. Okay. As a reminder, this is the principal component plot of the unadjusted data. And here is what the replicate matrix looks like. So I've created this M using the replicate.matrix function. And what I've done is I've passed it a data frame that includes the patient information and the brain region information. So any 2 rows of the data that correspond to the same patient, same brain region, those are going to be treated as replicates. Okay, so this ends up making a matrix that looks like this, and basically the way you can think of this is the first 3 rows, the first 3 observations in our original Y matrix all correspond to the same person, same brain region. So they are replicate set 1. The second set of 3 rows, those are, again, same person, but a different brain region, and so on. Okay, so this is what the replicate mapping matrix looks like. We pass that into our UV3, along with the spike-in controls, and this is the adjusted data that comes out. Okay, good. Here I'm going to do the same thing, except I'm going to set the averaging equals true option. When that pops out, okay, so I should go over here. In this plot here, there's 90 points, 3 for each tissue sample. In this plot, so I've set the averaging equals true option. In this plot, there are 30 points. one for each tissue sample. So in other words, here there's a point for each tissue sample. In the other plot, there's a point for each microarray. Okay. And again, you can see you end up with very nice clustering. The green are the cerebellums, and the blue and the red are the two types of cortex. Okay, so now I want to do something a little bit more subtle, and I think something that is really illuminating in terms of how all of this RUV stuff really works. And what I'm going to do now is, instead of plotting SVD plots, principal component plots, of all of the genes, I'm going to make principal component plots just of the genes from the X and Y chromosome. Okay. Let me ask you guys, if I do that on the unadjusted data, what do you think it will look like? How will it cluster? What's that? By the lab. On the unadjusted data set, yeah, it still basically clusters by lab. Okay, good. What might we want it to cluster by? What's that? Yeah, okay, so it's just the genes from the X and Y chromosome, right? We might want it to, we might be thinking that it would be good if it clustered by sex. Okay, here this is just different plotting stuff, but okay, now I've got the gender as the plot symbol, and I've got brain region as the color. Here I have done the adjustment, the RUV3 adjustment, using negative controls as the spike ins, and the replicates are the technical replicates. And what do we have? Well, actually, what it clusters by is brain region. Okay? What this is starting to get at is the important point that in any analysis, it's in some sense up to you what is wanted and what is unwanted. Depending on the goal of the analysis, certain things might be wanted, other things might be unwanted. So here we've done a very good job at removing the purely technical variation, but we still have what might be considered in some cases as unwanted biological variation. Okay? What this means is, and what we're going to see in the next couple plots is, that by changing what I use as negative controls and by changing what I use as replicates, I'm also going to be in some sense redefining what variation is wanted and what variation is unwanted, and I'll be ending up with a different adjustment. Okay, so here comes example 3. I'm going to do basically what I just did, except for now as negative controls I'm going to use housekeeping genes. And as replicates, I'm going to use all observations from a single patient. Okay? So by treating all observations from a single patient as replicates, so for patient 1, I've got as replicates the 9 different arrays from the 3 different tissue samples, but importantly I have different brain regions included in this set of what I'm calling replicates. Okay? Which means that I will end up adjusting away variation that is associated with brain region. Okay, so I now have a new replicate matrix. Now it only has 10 columns instead of 30. And so here I've got the first 9 rows. Those are all of the samples from patient 1. The second, those are all of the samples from patient 2, and so on. Again, I'm treating even different brain regions as replicates as long as they all come from the same patient. And when I do that, this is the adjusted data. It separates very cleanly by gender. Okay? So again, the high level, and I think very important point, is that, well, one, in any analysis, it's really in some sense up to you what should be considered wanted and what should be considered unwanted. And also that these RUV methods give you some way of addressing that in a somewhat natural way by what you consider to be negative controls and what you consider to be replicates. Okay, and I want to make a comment before I go any further on this point, which is that in this last plot here, where we get this very nice separation between the males and the females, there's more going on here than what you might think of as just regressing out brain region. So it probably feels like what I just did is, okay, well, I regressed out brain region. There's more to it than that. So here I'm going to give you some code that does quite literally regress out brain region. So it creates a dummy variable for the brain region. It does a regression. It takes the residuals of that regression. So essentially, you've regressed out brain region. And when you do that, this is the data that you end up with. Oh, I should have said, I'm sorry. Here, first, I've done the RUV3 adjustment with the housekeeping genes and the technical replicates. So I've cleaned up all of the technical variation. And then I manually regressed out brain region. And when I did that, here's what I end up with. Okay, so it's not very nice, clean clusters of gender. Now there's this outlier. I'm going to manually remove the outlier. That doesn't really fix things. It's still not a nice, clean clustering by gender. If I go on, now I'm looking at the third and fourth principal components instead of the first and second. And here I do see a clustering by gender. But again, this is in the higher order principal components. This is in the third and the fourth. It's not in the first and the second. So what's really going on here? What did I do different with this RUV3, with the housekeeping genes and with the biological replicates that is different than just regressing out brain region? Well, one is that by using housekeeping genes and biological replicates, that offers a much stronger adjustment overall that can adjust for all kinds of what we might think of as biological unwanted variation. A second important point that is admittedly not entirely relevant to this example, but could be very relevant for other examples, is that if I were to just go out and naively regress out brain region, I might take out biology with me if the biology and brain region are correlated. Okay. Whereas the RUV method, even if you have correlation between the unwanted and the wanted, it will only remove the unwanted. Okay, so that's the difference there. Shoot, that gets us right to 3 o'clock. I'm going to skip that. I might just, okay, I'll go about 5 minutes over. And I will introduce the Shiny app, just so that you can see it. Okay. So here I load the RUV library, I load the Shiny library, color picker that's needed for it to run, and then I'm going to load the gender data. I'm going to use the normalized data, and then I'm going to just run this RUV Shiny command. Okay, so I do that. It starts running. I'm in the Docker app, so I need to manually come out and come to here. But here's what the Shiny app looks like. Okay. So on the first page, it shows the gene expression data. That's the data matrix. You can look at it just to make sure that it looks sensible. Here's the row information. So I've got the different patients, their genders, the brain regions, which lab, and so on. And then also the column information, or the information on the genes. What type it is, its symbol, the chromosome, etc. Okay. one of the very first plots that you'll want to look at is a canonical correlation plot. So let's say that our factor of interest is gender, and we are thinking of using the spike-in controls as our negative controls. What this plot tells us, okay, so the horizontal axis here is k, and that's the number of principal components that I'm going to be taking of the negative controls. And the y-axis is the canonical correlation, actually the canonical correlation squared, of those first k principal components of the negative controls and this factor of interest gender. So what this is telling us is that, for example, if I take the first 20 principal components of the spike-in controls, and then I take the canonical correlation of those first 20 factors, the canonical correlation squared looks to be about 0.2 with the factor of interest, with gender. In a well-designed experiment, the canonical correlation will slowly increase at more or less a 45-degree line, straight up until it gets to 1 when you've made k as high as it can go. Okay. In this case, k stops at 33 because we only have 33 spike-in controls. Okay. So looking at this, what this tells us is that the spike-in controls seem to be okay to be negative controls. Let's compare that, for example. Let's say that our factor of interest was brain region, and we are considering using, well, let's just say, for example, all genes as negative controls. That obviously doesn't work very well. You very quickly have a very high correlation between the first couple principal components of the genes and the factor of interest. I forgot to mention, I'm sorry, let me go back, say I'm going to look at the housekeeping genes. The black is always all genes, the green is the genes that you've selected. Okay, so this is something that you'll want to look at the beginning of just about any analysis as a way of gauging do my negative controls appear to be correlated with my factor of interest. And in the next session, we'll look at some examples where we look at something like that. The final point that I'll make before I let you go for the break is that if you do see that your negative controls are correlated with your factor of interest, there's always two possible explanations, and you'll need to stop and think about which one is most likely. One possible explanation is that what you're calling negative controls actually aren't, and they're picking up biology. The other is that there actually is some correlation between the unwanted factors, some confounding between the unwanted factors and the biology that you're interested in. So when you do see that there is something like this, that there is very high correlation between what you're calling negative controls and your factor of interest, you need to ask yourself, do I think that do I still believe that these are negative controls? In the case of the housekeeping, I would say no. If I looked at the same thing with the spike-in controls, you'll see, interestingly, that the spike-in controls also have a pretty high correlation with brain region. The reason for that actually is poor experimental design. For some of the brain regions, they did all of one brain region on one microarray type and all of a different brain region on a different microarray type. And so this high correlation here is actually due to poor experimental design and actually is something that you want to adjust for. Okay, so I will let you go for the break, and maybe when you come back, I'll spend just the first 5 minutes showing a couple other things on the Shiny app, and then I'll hand it over to these guys. All right, thanks.